Carbon is regarded as the chemical basis of all known forms of life. Why is this so? The atomic number of carbon is 6, meaning that it has 4 valence electrons. This makes it able to form a maximum of 4 covalent bonds, making it very versatile in forming compounds with other atoms. Consequently, many of the molecules that make life possible contain carbon as a key component. Cells can synthesize substances through a method known as dehydration synthesis. As the name of this process suggests, a water molecule is removed to form a covalent bond between the reactants. The inverse operation to dehydration synthesis is hydrolysis, in which water is added to a molecule to break it down. There are four main types of organic compounds: carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates are the energy molecules of the cell. They are comprised of hydrogen, oxygen, and carbon atoms. Monosaccharides are the simplest carbohydrates that are monomers of more complex carbohydrates, such as disaccharides, made up of two monosaccharides, and polysaccharides, made up of many monosaccharides. The chemical bonds of monosaccharides, particularly glucose, contain energy that can be released through cellular respiration and used for the cell's functions. Disaccharides and polysaccharides can be broken down by hydrolysis into monosaccharides for this purpose. Similarly, monosaccharides can bind together through dehydration synthesis to store chemical energy. Some carbohydrates provide structural support, such as chitin and cellulose, as the monosaccharides can bind in a way that make it hard for enzymes to break it down. Lipids are a diverse array of hydrophobic molecules, containing many nonpolar carbon-hydrogen bonds. Lipids include triglycerides, phospholipids, and steroids. Triglycerides are comprised of a glycerol bound to three fatty acids. They are used primarily for energy storage, as they can be stored very compactly. For this reason, they are also used for insulation. Phospholipids are comprised of a glycerol bound to two fatty acids and one phosphate group. They have a polar head and a nonpolar tail. This causes them to bind to each other in a bilayer form, in which the polar heads are on the outside in contact with the aqueous solution outside, and the nonpolar tails are inside the bilayer. The phospholipid bilayer is the primary structure of cell membranes. Steroids are lipids with a carbon skeleton of four fused rings and are used for hormones and strengthening the cell membrane. Proteins are arguably the most important organic compound, as they fulfill a myriad of roles including support, storage, transport, signaling, movement, and defense. What makes this possible is protein's incredible diversity. Proteins are made up of monomers called amino acids joined together by peptide bonds. Since there are 20 types of amino acids, millions of different proteins with different amino acid sequences can be formed. Proteins have four degrees of structure: primary, secondary, tertiary, and quaternary. Primary structure is a sequence of amino acids. Secondary structure is whether the chain of amino acids forms an alpha helix or a beta pleated sheet. Tertiary structure is a complex 3D shape of the protein formed by folding back upon itself. Quaternary structure is the structure formed when two or more proteins interact. The unique and specialized structure of each protein enables it to carry out its function properly and effectively. Nucleic acids code for the proteins synthesized by the cells. There are two types of nucleic acids: deoxyribonucleic acid (DNA) and ribonucleic acid (RNA). DNA is contained in the nucleus of the cell and is the primary blueprint of the cell. RNA is a copy of DNA that is used to synthesize proteins from the DNA's code. The monomers of DNA and RNA are nucleotides. Nucleotides are comprised of deoxyribose or ribose, a phosphate group, and a nitrogenous base: adenine, guanine, cytosine, and thymine or uracil. The sugar and phosphate form the backbone and the nitrogenous bases form the rungs. This structure allows DNA to be replicated and transcripted. Nucleotides are assembled according to an existing template strand, thus forming a complementary strand. The chemical structure of mRNA also allows tRNA amino acid molecules to bind to it, thus permitting translation to occur. All three of these properties are very important for nucleic acids to perform their function correctly.